Okay, so we are recording this. So in this section, what we want to take a look at is we're going to get into the functional groups. And what you're going to find out is that the functional groups are clusters of elements that are attached to a larger molecule, okay? And they're going to give very repeatable and predictable characteristics. So when we look at something and we recognize, hey, that functional group is a polar component, that's going to help give overall polar characteristics to that molecule. If we look at something and say, you know, that's a donator of hydrogen. So if something was a donator of hydrogen ions, what would we classify it as? If it's going to boost the hydrogen ion concentration, we would say it's going to be something that drives up the hydrogen in a solution.
Acid.
Acid. All right. Thank you. Appreciate it. So we have certain functional groups that are going to accomplish that. We have certain functional groups that are going to do the opposite of that. So this is kind of understanding how the building blocks, just like yesterday's discussion talked about, you know, understanding the backbone and whether we had single or double covalent bonds throughout the system. So the functional groups are really quite critical in understanding how we build these more complex molecules. All right, so they definitely give properties that are distinct to the different organic molecules, and you know they're gonna have predictable characteristics that we're going to be able to go through there. So that's going to help us have a better overall understanding of what the properties of that larger molecule are going to have. Okay, so those functional groups we said were components of organic molecules that are most commonly involved in the chemical reaction component. So in many instances, they're towards the perimeter of those molecules, and they will give it unique properties. So pretty much everything that I just said in the intro on that is what's listed there. So if you take a look at this picture right here. Now, one thing that we're going to be able to recognize as we kind of move through is that we have these 4 fused carbon rings in both of these example molecules. And we talked the other day about the fact that the shorthand for this is just showing that each of these points is a carbon. And then when you have these double lines, it's a double bonds, okay? Overall, the general structure between this one and this one, yes, there's a little bit of a difference right here with the position of the double bonds and, you know, missing some double bonds. But the main differences that we're seeing are right here and right here, okay? And that changes the properties dramatically. Okay, if you're talking about primary hormone for women versus primary hormone for men, we're only looking at a few small components that are changing on there. So the point of understanding the different functional groups, and we have 7 of them. So we're gonna give you a quick run through these 7. Is that they are going to work to help us understand the overall properties of what you're going to see. And as a quick note that I'll put down here before we move into this, what we're going to find is that out of this grouping, this portion right here, so basically all with the exception of the methyl group, these are all going to increase the solubility in water. Okay, so we can just say that these are going to be hydrophilic. It's also going to indicate that they have polar components. All right, so these are going to all be polar, and this is the one exception to that group right here. Maybe ran out of the black ink. There we go. So the methyl group, as we're going to find out, those are going to ultimately be nonpolar in terms of their properties. Okay? All right. So what we're going to do is I have all of these things that we're taking right from the book. So we're going to zoom in and we're going to focus on those, and we're going to try to point some of these things out. Okay? Now, our first one is called hydroxyl. Now, this is showing that there's an attachment Okay, and it's it's not the same. Please don't make this be the same. Okay, this is hydroxide. This is an ion. This is not the same thing. This is hydroxyl, which is just saying that we have the O and the H. They're bonded onto the end of a molecule. All right, we see this a lot of times. The big thing that we want to know about the hydroxyl is that this particular thing is often referred to as an alcohol. Okay, the OH is going to make those molecules be classified as being an alcohol. So the hydroxyl group is polar. All right, and we would know that it would be polar because when you look at this picture right here, and we've got an oxygen and a hydrogen, we know even just from talking about water that there's a difference in electronegativity. So that oxygen is going to pull that electron pretty aggressively. So in many of these instances, this is going to form bonds with water, which means that it's going to be compatible, it's going to be hydrophilic, and it's going to help us dissolve things. Okay, so when we look at our carbohydrates in chapter 5, and we look at the structure of glucose or fructose or any of the other sugars, we're going to see that there's lots of these OH groups on there. So that increases solubility, and it makes it it gives it polar properties. Okay, so you definitely want to recognize the OH. It's going to help us dissolve sugars and things of that sort. Okay, so the second one, and actually, like I said, a lot of these relate to the lab. This one here is definitely going to appear in the lab discussion today. Okay, we have what's called the carbonyl, and if I take a look at what's happening with this one, this is a branching where you have a carbon with a double bond to the oxygen, as it shows right there. And there's two general categories of carbonyls that we're going to see. We have this particular designation where this one is going to be inside the molecule in terms of the overall structure, and they would refer to that as a ketone. Okay, so if it's inside the molecule structure, and then we have this one right here, and I don't know why it keeps popping off, but this one, it's on the edge or the outside. That's called an aldehyde. So here's my easy way to remember the difference between, is it a ketone or an aldehyde? The key goes inside the lock. In a ketone, it's inside the molecule. The hide is on the outside, right? The hide of an animal is on the outside. So the aldehyde is on the outside of the molecule. So if you have the carbon double bonded to the oxygen and it's on either end of the molecule, that's going to be an aldehyde. So today, when we start digging into the lab, one of the things that we're going to see is that some of our sugars are what they call ketoses. So that's telling us it's a sugar that has this kind of configuration within it. And some of them are called aldoses, which is a sugar that has the carbonyl on the outside. Okay, so just recognize that it's a carbon with a double bond. And once again, these things, oh, here it is actually mentioning that, the aldoses and the ketoses. And because they are going to give that polar characteristic, it's also going to help with the solubility. Anybody have any questions on that one? All right. So moving on, we have our next one is called the carboxyl group. Now, carboxyl is oftentimes, if you're looking at a formula, the way that they write that out is COOH. So they don't write it necessarily CO2H, even though there's two oxygens. It's oftentimes written as COOH. So you will see, for example, over here. And when we actually look at how they're going to bond and how they're going to form their structure, what we're finding is that you have your centralized carbon. Okay, one of the oxygens is going to have a double bond, which is what we're seeing right here, and then you have your OH component that makes up this portion. So yeah, you might look at it and say, well, we just saw that was an alcohol. But when it's part of this overall structure here, this is going to be called carboxyl. So we see carboxyl in a variety of different molecules. You'll see it whenever we start looking at the fatty acids, for example. You'll see a component on there where there's a carboxyl component. And the hydrogen, if we look at this picture right here, so this image is showing us acetic acid, which the name in and of itself indicates it's going to donate hydrogens. So when we look at this and we have the release of the hydrogen coming off of there when it's ionized, what that means is it's going to act as an acid because it can donate hydrogen. Okay? And it says that happens because it's a covalent bond between oxygen and hydrogen is so polar. So those are some examples. It's an organic acid. So when we're looking at a molecule and we're looking at an image and we say, all right, well, is this going to have acidic or basic properties? If you're seeing that it has the carboxyl making up a portion of that molecule in that area, you know, you're gonna be able to donate hydrogen off of that. Okay, so that's a common one, and it's definitely something that we will see as we kind of go through there and you know, examine some examples of that. Now, when we move to the next one, and like I said, all of these up to this point and all the way up to methyl, all of them are going to serve as examples of things that are going to increase solubility. They're going to be hydrophilic. So our next one is called the amino group. So the amino group is going to be NH2, and obviously their component found in the amino acids, as well as the carboxyl. So if we look at this example right here, this is telling us glycine. Here's my carboxyl, C, double bonded to the O, the OH, so that's a carboxyl group. And then here you can see you have your NH2. Now, when we look at this, the free hydrogens in many cases are going to go and they're going to attach to that. So if it removes the hydrogen out of a system, so anything that can pick up hydrogen is going to act as a base. So once they become ionized, they actually are going to become NH3 with a plus charge to that, which is what we're going to see right here. So N with 3 hydrogens with a slight positive to it. Okay, so when we compare that, when we're looking at the structure of an amino acid, and what do the amino acids build? Right, they're the building block of the protein. So we have our 20 amino acids. They're all going to be the same all the way through here with the exception of the function or the R group, which would be up here. Okay, so we'll see that here in a minute. So aminos will give you those characteristics, and they will act as a base. All right, so keeping with the theme of protein, we have this next group. You'll see the combination of the sulfur and hydrogen. This is our sulfhydryl group. Okay, now what these are going to do is when we take a look at something that is a protein, we're going to find out that there's more than, so actually before I give that away, what is a protein? Like what goes into a protein? So we know it's amino acids, right? Is it more than amino acids? Good. All right. So we have the mention of the bond known as a peptide bond. So we have amino acids bonded together in a very specific order. All right. Does that make a protein?
No, it depends on the protein shape.
Oh, okay, good. So the first level, thank you, I appreciate that. The first level is what we've described. But what we're going to find is, and this really is chapter 5, so that's not necessarily what we're doing right now, but we have primary structure, secondary structure, tertiary structure, and quaternary structure. So a protein is more complex than just a sequence of amino acids. Like if you get the amino acid sequence wrong, it's definitely going to be a wrong protein. Like there's no question about that. You could trace that back. What determines our amino acid sequence? Your DNA. So if there's a mutation in the DNA that has occurred, it's going to code for the wrong sequence of amino acids. So when we think about it, our proteins are what we see. It's like the manifestation of the DNA. It's what your body structure is built from. It's your enzymes, you know, so many different things. So you might have a mutation in DNA that eventually causes a protein to be malformed or take on the wrong shape. So there's a lot of structure that goes into a protein that comes in. Now, why am I talking about that right now? Is that part of that overall protein is that we have multiple structures that have to be kind of cross-linked. And one of the things that helps stabilize that cross-linking are the sulfhydryl bonds, the linkages, the sulfhydryl linkages. So one of the things that we find, and it mentions it in here, but if you think about somebody that wanted to straighten their hair, okay? So if you straighten your hair, or if you do the opposite, if you curl your hair, you have probably two strategies to go about doing that, right? How would you straighten your hair? Heat, right? So what do you think you're doing? You're breaking some of that cross links and you're forcing it to reform into a way that is maybe not the natural configuration. So somebody that maybe has naturally curly hair, which there's people in my house that have that, not me, but if they straighten their hair with one of those straightener devices, it doesn't stay that way forever, right? Because your DNA coded your amino acids to be in a certain sequence, and your proteins in something like your hair are going to have a certain combination and building blocks with certain types of sulfhydryl linkages there. So you can kind of force it to do that way. You can change that structure with chemicals. So sometimes people will use chemical straightening things or chemicals to curl something. But after a period of time, it goes back to what your DNA is coding for. Okay? But sulfhydryl, just understand, and then this is true for pretty much all of them. I wanted to point this out because sometimes when we look at something, it might throw you off. This is just showing us, all right, this is the attachment point. So if the attachment of the sulfhydryl came to the right of the overall molecule, you'd see it listed as the bond and then the sulfur hydrogen. If it happened to be on the left side of the molecule, you'd see, okay, here's the bond connecting to like the larger molecule, and then here's the sulfur hydrogen. So the sulfur portion of it is what is attached to that bond that's connecting to the other component of the molecule. So this designation and this designation really are different. It's just showing you where it's at positional for that particular scenario. And you could see that with the other ones that we did as well. All right. So that is critical in our study of protein. Okay, moving along, the phosphate group, when you start to think of phosphate, we can see right here PO3, but in many instances, it's going to go through there and it's going to have a greater pool, and ultimately it's going to become ionized and change, which we will see here in just a second there. So phosphate is going to be part of, in many instances, our energy molecules. So when we think about adenosine triphosphate, ATP, main energy molecule, guanine triphosphate. So a lot of times what we're going to find out is that these things are going to have the ability to react with water, and they are going to release a quantity of energy. So we have a unit, chapter 8, we get into quantifying how much energy you get from a mole of ATP. And then we could trace it up and say, all right, well, if we took glucose and then we metabolize that, how much energy can we get in the form of ATP? So, but we understand that in these cases right there, it says they're gonna have a negative charge to that. So in this case right here, we can see that This, a lot of times, may be written as PO4 because you have the 4 oxygens that are going to be part of that overall linkage right there. Okay, but just make sure you understand that that's going to be the one that's going to be most often associated with, the release of energy. All right, and as we go through here, our last one that we want to talk about is called the methyl group. Okay, so methyl, out of everything I went over today, is the exception to the rule. So when we look at methyl, this one here is not going to increase the solubility. Okay, methyl is going to be the base unit of a carbon with 3 hydrogens wrapped around it. And one of the things that we talk a little bit more about later on in the course is the role that this plays in gene expression. Okay, so we found that the attachment of methyl groups to certain genes in the DNA have the ability to silence them. So if you think about this, you've got your DNA code, and within that DNA code, you've got segments that are the genes, and our DNA is the same in all of our cells, but the proteins that we make in those cells are unique to the cell. So you're not making digestive enzymes in areas that are part of your respiratory system, for example. So one of the things that happens is that there are processes of silencing and initiating different genes. We also found that chemicals in our environment that contribute methyl can actually shut down genes. So to do a really, really simple example of this is if I were to say, all right, I have my DNA, and if I were to take some string and just kind of wrap the string around my fingertips, okay, so almost like I had yarn representing the DNA, and there are proteins that are associated with DNA. Does anybody remember what the proteins are called? Start with an H. Histone proteins? Okay, that's fine. We'll get to that later on. But the idea is, if my DNA was loose, and my fingers, you notice I'm kind of extending my fingers apart as much as possible, I would have sections of DNA that would be accessible by the ability to transcribe, to make protein out of that. When we add methyl groups to those proteins, and I'm representing the tips of my fingers as like those histone proteins, it does this to the DNA. It closes the DNA down. So if I were trying to bring the transcriptional machinery to get that code to start the process of making a protein, it's shut down. So we silenced it. Now, interesting area of biology that's emerging is called epigenetic inheritance, which is saying that the things in our environment, the things that we've eaten, and even crazier, some of the things that our parents, and even as far back as our grandparents, that were exposed to or consumed, can influence 1 or 2 generations down the line as far as expression of things. So it's a whole area that we're going to focus on later on. But for right now, the main thing is just be able to recognize what a methyl group is and the fact that it is going to not increase the solubility. So it's going to be more of a hydrophobic component. All right. Does anybody have any questions on those? Okay, so I'm going to stop the recording and then we'll move on to the lab.